These are the recorded lectures for Chapter 20 on viruses, bacteria, and archaea. And the sections that we'll cover are, Section 1 is on viruses, viroids, and prions. And then Section 2 is on COVID, basically. Section 3 is on just reminding you, it's like a review on prokaryotic cells, reminding you that prokaryotes don't have a nucleus or membrane-bound organelles, and that prokaryotes are always unicellular. The two domains of living things that consist of prokaryotes are bacteria and archaea, so we'll cover sections 4 and 5. Now, we already covered the section on viruses, so I'm just going to review very quickly. Make sure that you know some of the common diseases that are caused by viruses. You'll need to know that they're caused by viruses and not bacteria. So, of course, HIV is a virus that causes AIDS. There's genital warts caused by the human papillomavirus. There are several herpes viruses, genital herpes, and also like fever blisters, cold sores. Those are also herpes viruses. There's the mumps, measles, chickenpox. These are all caused by, there's childhood diseases which we get vaccinated for. There are respiratory diseases like the common cold, the flu, influenza, and of course, COVID, which is caused by the SARS coronavirus. Warts are caused by a virus. Shingles is caused by the same virus that causes the chickenpox. There's smallpox that we get vaccinated for. There's mononucleosis. So just try to review over that list and make sure that you know diseases which are caused by viruses and not bacteria. And then we have the structure of a virus. A virus is always going to have at least a protein capsid, which is the outer coat, and then it's at least going to have DNA or RNA in the center of that capsid. Some viruses will also have an envelope outside the protein capsid, and then some viruses will also contain their own enzymes to help replicate their DNA or RNA, or for processes like transcription, translation, that kind of thing. So they're always going to have, let me highlight the two parts that a virus is always going to have: a protein capsid and a nucleic acid core, which will be DNA or RNA. And then we looked at some images of typical viruses, and I reminded you to make sure that you understand a virus is an obligate intracellular parasite. That means that obligate means that it must live inside, or it must enter a cell in order to reproduce. It cannot reproduce outside of a living cell. And we went over the stages of the viral life cycle, which are attachment, penetration, biosynthesis, maturation, and release. There we go. All right, so you can see this is the lytic cycle for a bacteriophage, and bacteriophages only infect bacteria. So here's your bacteriophage. You can see the green protein capsid, and then you can see the nucleic acid inside, which is that purple wavy strand. So what the bacteriophage does is, first it attaches to the cell wall of the bacteria, and that's the attachment stage. Stage 2, penetration, it injects its DNA into the bacterial cell. Stage 3, its DNA is used to make more DNA, more viral DNA, and also to synthesize new protein capsid, and that's called biosynthesis. Maturation is when the bacterial cell actually puts the components of the virus together and completes the production of new virus or new bacteriophage. And release is when those viruses emerge from the cell, either destroying the cell or harming it in some way when they are released. Probably they destroy the host cell. But now these new viruses can infect new host cells. So that is the lytic cycle, which is the active cycle. Sometimes the bacteriophage will go into the lysogenic cycle. After penetration, the viral DNA will incorporate itself into the bacterial DNA. And then there will be a period of time where nothing will happen. It will just be like a dormant time, a waiting period, and then something will trigger that DNA to start being, trying to think of the word. Something will trigger the DNA to begin, for its genes to begin synthesizing more DNA and protein synthesis of the viral capsids. So it will remain in a stage called integration, where that viral DNA has integrated into the bacterial DNA. And that is called the lysogenic cycle. It's a dormant stage or a latent stage. So the virus will remain latent or dormant for a period of time. Something will trigger the lytic cycle to begin, and it will enter the biosynthesis stage of the lytic cycle. Something like stress, or sickness, or in the case of a bacteria, stress would be like lack of nutrients, lack of water, lack of space to grow, that kind of thing. Retroviruses such as HIV are viruses that contain an enzyme called reverse transcriptase. They are RNA viruses, and what the reverse transcriptase does, if you remember, transcription is where DNA is converted to messenger RNA. So if we reverse that, RNA is going to be converted to DNA, in this case, cDNA. This is called reverse transcription. Once this happens, that cDNA can now be used to replicate new HIV viruses, new virus, So those stages are shown here. The main thing is that there is a stage that comes before, after attachment. HIV is an envelope virus, so it attaches and the whole entire virus enters the host cell, which is one of our white blood cells. A human white blood cell called a helper T cell is always the host cell for HIV. So now the entire virus enters, and then the third stage that occurs before biosynthesis, and it's the stage where the RNA inside the virus, the RNA is transcribed into DNA. It's called reverse transcription, and the virus has this little blue dot is the enzyme reverse transcriptase, and the virus uses that to convert RNA to DNA, and then the DNA can then be used to make more viral RNA, and it can be used to make more viral components, and so synthesize new virus. And then the envelope forms as the virus leaves the host cell. The envelope forms around it from the plasma membrane of the host cell. Now, moving on, we also have these disease-causing chemicals called viroids. Disease-causing pathogens. Anything that causes disease is called a pathogen. So in addition to viruses, we can have viroids, which are simply naked strands of RNA. They're responsible for causing many crop diseases, so plant diseases. And then we can also have a pathogen called a prion. And a prion is simply a protein with a contagious tertiary structure or three-dimensional structure. So it's a protein molecule that has some kind of mutation, I would say. And prions cause TSEs, which are a type of neurodegenerative disease. In other words, the nervous system, nervous tissue in the brain begins to degenerate and until the person is dead. So it's untreatable and fatal. These are terrible diseases. But some examples of TSEs caused by prions are, and that just stands, what is TSE? TSE stands for spongiform encephalopathy. I can't remember what the T stands for. Not bovine, but let me see if it says it somewhere. No. Great. I can't remember what the T stands for, but the S and the E stand for spongiform encephalopathy. If it's BSE, then the TSEs can occur in humans, basically. And then BSE is a cattle disease caused by prions. So mad cow disease can actually occur in cows, and also it can be transmitted to humans if they eat the brain tissue of the cows. There's a section on the flu virus, and it just explains. You do have a question in your Connect assignments where you have to know this, but it explains that the flu virus has these spikes, glycoprotein spikes on the surface of the envelope, and those protein spikes are either H or N spikes, and the type of H spike and the type of N spike determine the version of the flu that you have. So we have, for example, H7N9 and H5N1 are subtypes of the flu virus, and they just name the protein spikes, the H and the N protein spikes. So what the H spike actually does in the flu virus, it allows the virus to bind to its receptor on the host cell. And there are 16 different types. So you can have H1 all the way to H16. And the N spike has a point on it, and it attacks the host cell's plasma membrane. So it enables the flu to enter the host cell. I'm sorry, I said it wrong. It enables the flu virus to leave the host cell, which does kill the host cell. And there are 9 different types of N spikes. So you can have N1 all the way to N9. And this is a slide which shows us how the bird flu and the human flu could mix and cause a version of the flu which can infect birds and humans. So what happens is, for example, in situations where humans are in contact, really close contact with a lot of birds that maybe have respiratory infections, and their mucus can mix somehow, when they're in really close contact, for example, turkey houses and chicken houses, people who work in those, in that case, the bird flu virus can actually integrate and mix and reassort. It's what's called reassortment. But it can mix with the human flu virus and create a combination bird-human virus that can infect both the birds and humans. Now, section 2, we're just going to briefly cover emerging viruses are either new or they're previously uncommon. So a very good example of an emerging virus, just back in 2019, we all experienced COVID, which is a coronavirus that causes the like cold, like respiratory symptoms. There are other ones listed here. The bird flu, avian influenza, or bird flu is another example. So what can cause the emergence of a virus? The virus can extend its range. And we saw that happen with COVID. It started not in the United States. I can't remember. I think it was China. But it definitely started in Asia. And then it extended its range to the United States and the rest of the world. The example that's given here is the West Nile virus was transported to the U.S., and it actually started out in birds and mosquitoes, and then it ended up being able to infect humans. So when the virus extends its range, makes it possible to infect a larger number of people, and then also if the virus mutates. So genetic mutations and the extending the range of the virus infecting many more areas of the world than what it originally did, those things can cause a virus to become an emerging virus. The Zika virus is spread by mosquitoes. And if the Zika virus infects the fetus, in other words, if it's spread from the mother to the fetus when she is pregnant, it can cause microencephaly. Microencephaly is this small head size, microencephaly. And they would have definitely various side effects, but they aren't good, And this just shows you the distribution of the Zika virus in the United States. Not in humans, but in mosquitoes. So it's going to, of course, be more in the south because it's warmer. This is an illustration of some of the emerging viruses. We've got 2003, there was a SARS virus similar to the COVID-19. And then we had 2019, SARS-CoV-2 is the COVID-19 virus. We've got avian influenza, 2003. Got some that emerged in the 90s. MERS emerged in 2012. The West Nile was 1999. You have to Google this one, but chikungunya was 2014. So these are just different emerging viruses that we're aware of at the moment. This is a picture of the SARS-CoV-2 virus, the one that causes COVID-19, and it shows you the proteins on the surface of it, which are the S protein, the M protein, and the E protein. These are the glycoprotein spikes on the envelope that enable it to attach and enter the cells in our respiratory system. All right, I'm going to stop here, and I will start the next recording with section 20.3 on prokaryotes.